Urmila Kulkarni Kale, second by Devasis Mohammed, and third by Sudipto Saar. Okay. So first topic by Dr. Urmila. He is well known to the persons who are working in biotics because she has long career in bioinformatics. She did the PhD from Pune University. In the same university, she is information scientist, and she is contributing to in the area of biotics from long time. Both means in terms of the education, service, and research. In all three areas, actually, Bioinformatics Center at Pune is very popular actually, and contributed a lot in the field of bioinformatics. And she is one of the major contributors. And as far as her research is concerned, she normally works in the evolution of viruses using the population genomics. And she is responsible for developing number of databases, softwares, algorithms over the years, and more than 25 years of teaching experience. The number of students who are in the biotics, they pass out from the Pune University. Actually, a lot of people have passed out from Pune University. Actually, they are continuously. She is a elected member of Maharashtra Academy of Sciences and editorial board of number of journals, including Journal of Infection, Genetics and Evolution. So I will not take much time between you and Urmila. So Dr. Urmila, you are welcome for that.
Thank you, Dr. Raghava, for this generous introduction. And thank you very much for having this conference organized. It is the most appropriate time to have a conference of this kind where we talk about health informatics and your initiative to develop a large number of databases servers in the area of health informatics, along with your initiative to organize this health informatics conference to consolidate contributions of Indian scientists is really commendable, and heartiest congratulations to you. Thank you very much for inviting me to deliver this talk. Without investing too much of time, let me begin. And at the onset, let me also thank Neelam for operating my slides because technology is good, we use it, but then because of some power surge, looks like my video cable burnt off just before I was about to load my PPTs. So I requested Neelam, and she is generous enough to operate my slides for me. And thank you, Neelam, for that. And thank you, Raghava, to accommodate this last time glitch as well. So let's talk about, since we are talking about health informatics, let us start with the concept of health. And you would know that the concept of health became inclusive and broader with UN's Sustainable Development Goal of One Health. So we are now thinking of the health of the universe, which includes, humans, animal, plants, interactions of, species of their kind amongst themselves, interactions of, their microbiome, viromes, and so on and so forth. So it's a very interesting time indeed to look at the health from a very, very inclusive and a broad perspective. And it's very interesting that, we are talking about the health, one health at one end of the pyramid. And, at the other end, you have billions of these species for which, you are going to make the health considerations. And just like we use the Google Earth, to zoom in and to zoom out, we talk about health at physiological level, at cellular level, at molecular level, and then we use reductionalist approach to study various aspects and components of health, and then we use informatics to rebuild the connection and to make discoveries with respect to health of the systems around us. Next, please. Neelam. Yeah. When Raghava sent this invite for this talk, and in particular, I decided that I would talk about immunoinformatics. And in the context of One Health, the concept that came to my mind was the concept of multiverse, or parallel universe. So every organism is complex in itself, be it human, be it an animal, be it a plant. And then we are talking about human, animal, human, plants, animals, interactions, and so on and so forth, in order to make understanding with respect to their health. Host in itself, we talk about and study physiology, the phenotype, the genotype, the immune system. Then we have the inherent, microbiome and inherent viromes for every organism, every species. And then there are some transient pathogens which also come and go. And in this perspective, then I thought that multiverse probably is the most appropriate kind of a concept under which we can relook at, immunoinformatics. So host and pathogens, they have been around for many, many years. They have independent existence, and they also coexist. The arms race for survival of both the host as well as the pathogen enabled, the evolution of immune repertoire of the host and the evading mechanisms of the pathogens. So this co-evolution has taken place. And when we talk about multiverse, let us say a pathogen is an universe in itself, and a host like human is an universe itself. So when they interact, when they meet at the interface, what we see is the immune system. And that is why I thought that when we talk about immune system and immunoinformatics, it is at the interface. And what kind of mining can we really do at the interface of host and pathogen interaction using immunoinformatics is what I'm going to discuss in next few minutes. Next, please. So I believe that immunoinformatics is informatics application to immunology, which is a very, very basic science. And immunology is a basic science. Immunoinformatics breaches basic science like immunology with the translation science, wherein we are talking about product development using synthetic biology. So when we talk about immunology, the binary nature of immunology is very, very evident, that you have innate or acquired immune system, you have humoral and cellular immune response, you have B cell mediated immunity and T cell mediated immunity, you have a huge repertoire of antibody. So antibody diversity, the combinatorial involved in the genetic recombination actually made informatics amenable in immunology. And of course, there is a component of hypersensitivity, which we are not going to deal in this lecture, but we are going to restrict our discussion to host pathogen interactions. And when it comes to the immunoinformatics, it's a data science where we are talking about diverse type of data that we are dealing with. We are talking about databases, algorithms, workflows, and immunology with systems biology. A variety of data is being generated, from single cell studies to organ and species level studies. So you have enormous data developed, and then I have used this beautiful illustration licensed from the Gapping Void, only for the purpose of academic inclusion in the PPT, that you have data which is represented as individual dot or points, and then you are translating this data into information, information into knowledge, and when you are able to connect to apparently very distant points, you gain an insight using algorithmic pattern matching, machine learning, processing, and then what you come up with wisdom that can be translated into socially useful products which deliver impact onto society. So if we talk about data about immunoglobulin related genes or antibody diversity or B cell T cell epitope, they are just the data. You are transforming that data into information and knowledge, wisdom, and translatable products. When I talk about, I'm referring to the development of vaccines or development of diagnostics or development of therapeutic antibodies. That kind of application one can think of in terms of translatable products. And this whole field of translation is growing with about 23 to 25% of CAGR every year, and it is projected to grow at even a higher rate. And this current pandemic, the SARS-CoV-2 has actually pushed everyone, the entire global scientific community, to come together to sort of collaborate at the level of the unprecedented level, where the data generated by individual scientists, the papers published by various groups are made available without any restrictions in the public domain. This SARS-CoV-2 genomic data is available, along with so many other types of data, including the immune escape variants and a lot of other experimental outcomes. So you can probe these data that is made available in public domain with the informatics or bioinformatics technologies and algorithms, and can think of understanding that data to come up with some kind of interesting insights in the data that can be used for the purpose of discovery and development. Next, please. So we at Bioinformatics Center at Pune University have been studying and looking at viruses. We are intrigued by these pathogens, very, very small pathogens. And we have been studying for many, many years as to how these pathogens infect host. How do they cause disease? How do they learn to jump species? Okay, how do they spread and evolve? How do they escape the immune system? And how resistance is developed in terms of drug resistance or immune escape from vaccines and so on and so forth. So these are some of the intriguing questions that we were trying to address across a variety of viruses. And I mean, that like for microbes you have bacteriophages. For animals, there are several animal viruses. I have given an example of FMD. For humans, it is HIV or SARS-CoV-2, the pandemic that we are experiencing, and for plant, tobacco mosaic virus and other. So you have the huge array of viruses that infect the variety of species in each of the 5 kingdoms of life. And we have been studying the diversity of these viruses using bioinformatics methodologies. So we in the process we look at the data, we analyze data, And viruses are so small. There are no big consortia for their sequencing or anything like that. But these are sequenced by individual virologists, put into public domain, and the data is available for analysis. Of course, SARS-CoV-2 brought the disruption of its kind, and then the pandemic, something that began as an outbreak in Wuhan province of China in the last week of December, grew by leaps and bounds and out of proportion, and brought the entire world under the stress of COVID-19 disease, and large number of sequences for epidemiological purposes have been obtained. Data is stored in the GIS AID and several other databases. We have also looked at that data. I'll talk about it a little bit, but this is the backdrop for our research at Bioinformatics Center in Pune University. Next. So our journey began in immunoinformatics, way back when I had joined as a student, and in fact that was a very interesting project on Japanese encephalitis virus, where we were making an attempt to develop a synthetic peptide vaccine. And obviously, Japanese encephalitis virus, like dengue virus, is a flavivirus. It is transmitted by mosquito. Every year you have outbreaks in various regions of India, and we were looking at synthetic peptide vaccine development. And as a initial study, I looked at the envelope glycoprotein, which is the surface protein of this Japanese encephalitis virus, and tried to predict the epitopes. So that time, a few epitope prediction methods, the sequential epitope prediction methods were available, like Hop and Woods, Parker's method, or there is a very elegant method developed in our own group, Kolaskar-Tongaonkar method. So and then there were methods developed globally. So I started with prediction of epitopes for envelope glycoprotein, and those sequential epitopes or antigenic determinants were predicted. I tried to look at the consensus prediction amongst the various methods, made my own understanding. But then if one is trying to develop vaccine, then finally when vaccine is given to the host, the immune repertoire or the antibodies needs to be generated, and those antibodies need to go and bind to the antigenic proteins in order to neutralize the virus. So that's the basic understanding. Then I started asking these questions as to whether these epitopes that are predicted by various methods, let us look at the location of these epitopes and how and where are they present on the envelope glycoprotein. And then I realized that the three-dimensional structure for envelope glycoprotein wasn't available, so I homology modeled the structure, and then looked at using sequence alignment approach, looked at the conservation and variation amongst the related viruses. So you can make this comparison amongst the strains of the same virus or related viruses, and then you can ask these questions: how similar is similar and how different is different? Because if you are a modeler, 35-40% of sequence similarity is good enough. But if you are an immunologist, so even 97% of similarity and 95% of identity, those few differences in the amino acids, the immune system has evolved at an exquisite level of specificity where such small differences are also detected. So if you inject a chimpanzee protein into human and human protein into chimpanzee with 4 or 5 amino acid differences, both the immune system recognize these injected molecules as non-self. So it is at that level of specificity, one is trying to look at similarity and differences, and that is where we made our understanding. Next. So you would need, very different amino acid and the charge distribution in the antibodies in order to neutralize the viruses or viral strains. And then this was the first attempt in the area of immunoinformatics for me, where I tried to look at, on the left-hand side, what I'm showing you is this Japanese encephalitis peptide design project, where we use MD simulation approach to build the structures of the peptides. We looked at the conformation, and we came up with a chimeric peptide model where we stitched the B and T cell epitope as shown here using a linker, and that linker also was drawn from the Japanese encephalitis virus. And on the right-hand side, I'm showing you a snapshot of one of the first algorithm for conformational epitope prediction. So conformational epitopes are nothing but antibody binding sites on proteins. And these binding sites, if you try to look at, it typically constitutes of, a set of linear antigenic determinants come together due to the folding of polypeptide chain. And then we developed this algorithm for which we use 3D structure data in an explicit fashion. So we use the accessible surface area in an explicit fashion. Until then, all epitope prediction algorithm, especially the B cell epitope prediction algorithm, linear ones, use the propensity values for accessibility, flexibility, and other parameters. So this was the algorithm, which is first algorithm for prediction of conformational epitopes or binding site, that was developed here at Pune as part of my PhD project with Professor Kolaskar. And then this proved to be very, very helpful in all our downstream projects. Can we go to the next slide, please? All right. So, we started deriving rules for knowledge-based predictions for the conformational epitope prediction using, about 22 co-crystal structure complexes that were available in the PDB database back then. And then I had studied these interaction interfaces between antigens and antibodies and characterized those surfaces in order to generate rules. But, by the time I came up with the algorithm, I had this huge data pertaining to antigen antibody complexes curated from PDB. Then I thought that probably it's a good idea to have it deposited in the database. And then we came up with this antigen antibody interaction database, wherein, we provided data at the atomic level details. We talked about what are the amino acid residues of epitope that interact with the paratope of antibody, what are the types of interactions, how many and where are hydrogen bonds, where are salt bridges, and so on and so forth. And we kept on updating this database, as well as refining our rules for epitope prediction. So we came up with a few versions of conformational epitope prediction server. So this is how these two projects went hand in hand. Next, please. So this particular understanding, I got a beautiful opportunity to apply this theoretical knowledge to a very, very practical and socially useful situation when Serum Institute of India. Well, you all must be aware of Serum. They are the world's largest manufacturer of vaccine, and the MMR vaccine developed by measles, mumps, rubella vaccine developed by Serum is given to every 5th child in the world. And even in the SARS-CoV-2 pandemic, they have come up with this Covishield vaccine, which has been licensed in India and certain part of the world. So Serum came to us saying that, the problem statement was like, some of the strains co-circulating in European countries are escaping neutralization, when population was given the MMR vaccine. And can you use informatics and immunoinformatics based or modeling based analytics in order to provide us with an insight so that we understand what's happening at the molecular molecular level, and if we can come up with certain interventions that can help us improve. So by the way, the vaccine is more than 98.5% efficacy. So it's a very good vaccine. So what we did, we looked at a set of mumps, isolates, and here the hemagglutinin neuraminidase is the antigenic protein. So we looked at the protein from the vaccine strains, from the wild type strains, and we predicted using homology modeling the three-dimensional structure for 4 proteins of hemagglutinin neuraminidase coming from wild type strain that was escaping neutralization, and 3 vaccine strains which were used by Serum and other manufacturers. And then this simple analysis pertaining to the amino acid substitutions. So when we plotted the data coming out from variable sites from multiple sequence alignment onto the 3D structure, all these mutations, they threaded very beautifully on the surface of HN protein, just like we thread the coconut. So and out of 28 mutations that were observed in HN protein, 24 were present on one surface, and then using that analysis, we sort of identified a major neutralizing epitope for the mumps virus purely using the computational approaches. And then we studied what are the mutations in the wild type strain that was escaping neutralization and the wild type strain. We realized that some of the mutations were very critical, including a mutation where a glycosylation site is lost. And this analysis helped us to put forward this very elegant interpretation for the escape mechanism way back in 2007. So since this project was sponsored by Serum Institute, initially they said that it is going to be a contract research, non-disclosure, etc., etc. But eventually, they sort of wanted these results to be published, and then we came up with this publication in the Virology in 2007. But next, please. But science is all about continuum. So this project sponsored by industry introduced us to a very beautiful concept, that these viruses, they are the continuum. They keep evolving, they keep changing. It's not a deliberate mutation or a change. Certain mutations happen in the process of replication. It is the infidelity of the RNA virus replication that these errors happen. But and most of these substitutions, most of these mutations are discarded in the process, but a few survive. And those mutations which provide fitness advantage to the virus propagate more, and that is how you have the new types, new genotypes, new serotypes come up. Just like we have witnessed emergence of Delta in India as far as SARS-CoV-2 is concerned. So this project from Serum provided us an outlook to look at viral population in terms of ensembles of genotypes and serotypes. And then when we started probing that area, we realized that when there are outbreaks, epidemics, or pandemic, how are these viruses typed or tracked? So phylogenetic analysis is the core of such a typing, and the alignment-based phylogenetic analysis, either involving individual genes or genome, is very, very time-consuming. You have to have reference data set. You carry out multiple sequence alignment. Then you look at bifurcating tree topologies. Look at the bootstrap values and the confidence scores for the phylogenetic trees, and then make your conclusion on the basis of proximity of clustering of unknown with the known, and you assign the genotype. And mind you, every time a new isolate is tracked from the environment or sequence from a patient, you have to repeat all these steps. And that is where, we thought about coming up with a method that's alignment free. So, can we develop a method for typing of viruses by eliminating these repetitive steps of having to carry out alignments? And this became one of the major areas. So at one hand, we are studying the host and the immune repertoire of the host. And in the second component, we are looking at viruses and viral population, and how do these viral populations interact and interface with the host. So that became the major research area. Next, please. So this was made possible because of a wonderful collaboration with my colleague Dr. Mohan Kale from Department of Statistics. So this return time distribution is a concept in stochastic processing, and we applied it for sequence analysis. So how does it work? So basically, it's a K-mer-based approach where we try to look at the words and how do they repeat, with how much of gap do they repeat. So for example, if you take a word size of just 1 and you have a representative genomic sequence, then we calculate RT or return time as time required for reappearance of a particular state. For example, I have shown, character A in the red color, and first occurrence of A at the 3rd position to the second occurrence of A at 5th position, there is a gap of 1 character, that is C. So A reappears after a gap of 1. Then again it reappears after a gap of 1, and then reappears after a gap of 0. So this character string can be converted into a numeric vector for the return time of alphabet A. Similarly, you can compute return times of T, G, and C if your word size is 1. If your word size is 2, then you will have 16 such return times to represent 16 nucleotide combinations, and then you can imagine the rest of the combination. All right, so you compute the frequency, and from frequency we derived very, very simple parameters, that is average and the standard deviation. Next, please. And then we use this average and standard deviation as our main attributes for computing distances between the strings of sequences, and the strings can be individual genes or can be individual genomes. So I have shown you an algorithm here. We can't get into the details of that algorithm, but basically you start with input sequences. Then you determine the word size, you compute the return times for K-mers, and then derive them. You derive the average and standard deviation. Then we used simple Euclidean distance to derive the distances amongst the sequences, and use simple NJ tree for drawing the tree, and tree visualization software for interpretation. So this is in a nutshell, this algorithm is all about. Next. This is the graphical representation from sequences to the phylogenetic tree that can be generated using this algorithm. Next. And this is the interface for the algorithm. I have given the URL and our paper in molecular biology and evolution for your ready reference. All right, we go to the next slide. So by then, we had come up with this pipeline, which is shown in extreme right in green color, which is alignment free. So for the classical molecular phylogeny analysis, the distance based and character based, which are shown on the left hand side, we added a third vertical, but then it has not solved. Next, please. It has not solved our problem of typing, and there was a gap. So we thought that now for this our basic algorithm of return time distribution based alignment free algorithm, we have to develop the wrapper which are dedicated, which are knowledge based pertaining to individual virus. And then we came up with virus specific typer servers, which I'm going to show in the next slide. So this wrapper we developed, wherein we have the reference data set, we pre-computed certain distances and use those distance values and the confidence interval for assignment of the genotype or the serotype. So now, next please. Using this pipeline, we developed several servers, but I would like to just give an example of one server, which is a human rhinovirus, again a common cold virus, which belongs to family Picornaviridae and genus Enterovirus. So, here we have used both the nucleotide and the protein sequences of VP1 protein. And in case of VP1 protein, K was just 1, 100% sensitivity and specificity. We could, assign serotypes HRV species. So HRV, by the way, are common cold viruses. There are 3 species, HRV A, B, and C, out of which C is non-culturable. So very, very challenging for typing using classical virological methods. Instead, it can be sequenced, and we provided a method for typing using sequence. So that's the salient feature for this particular server. And there are more than 140 different serotypes and genotypes known for this HRVs, and this typer made the job of virologist very easy. Whenever you have a new sequence, you copy, cut, paste the sequence in the server, click on the submit button, and the server would give you the type assigned. So, and again, for every server, we have the papers published alongside. Next, please. So using this philosophy, we expanded it, starting with, Mumps virus typer, learning from the Serum project, to the HRV typer. Then we extended this method for bacterial typing using MLST, and several servers have been developed. So these servers are available in the public domain, and you can use it. Several people all around the world, use this server for the purpose of typing. Next, please. All right, so we then had a project. All this knowledge that had gained, we thought that we can apply it for the purpose of new development and discovery. And we had this DST sponsored project in collaboration with NARI, Dr. Aarti Manes and Dr. Madhuri Thaka's group at ICMR NARI Pune, where we tried to look at, these are two different projects basically. With Dr. Aarti Mane, we looked at the genomic diversity from the Indian population for the human papilloma virus 16. It is one of the high-risk type virus. And this morning, Dr. Amit Datta was talking about, papilloma virus and the cervical cancer and the kind of elegant studies that their group has performed. So this was one project to track the genomic diversity and to correlate the mutations with the functional attributes. And the second project that we started and we are still working on is development of therapeutic vaccine for HPV 16. So you have the two types of vaccines, prophylactic and therapeutic. Prophylactic is a major category, that we come across, but the therapeutic vaccines are expected to be a good intervention, especially in case of human papilloma virus, because they are thought to reverse the grade of the disease. So CIN grade 1, 2, 3. If a patient is in grade 3, then she can be transformed from grade 3 to 2 is the basic premise for which world over several laboratories are trying to develop therapeutic vaccine. And this development of therapeutic vaccine is totally in silico. Here the theory came first, Next slide, where we came up with the pipeline and protocol for development of vaccine. I guess HPV, everybody knows it's human papilloma virus. There are 13 high-risk type papilloma viruses, including HPV 16, 18, 33, and so on and so forth. So they cause the cervical cancer. And HPV infection is prevalent in developing countries, including India. CIN, the grade status becomes very essential parameter for monitoring, and prophylactic vaccines are available. They need to be given to the men and women, the boys and girls, by the age of puberty. Nevertheless, therapeutic vaccines are being developed with an objective to reverse the grade of the disease. So vaccine targets are like L1 protein or oncoproteins like E5, E6, and E7. Next, please. So with this genomic sequencing, we identified those critical mutations that were observed in Indian samples, and we also came up with the typer server for HPV. I'm showing here the phylogenetic tree for HPV, and all those entries of coming from Indian patients. How are they clustered? And then, here again, there is a slight deviation from the vaccine project, but, HPV typing, earlier it used to be carried out on the basis of L1 gene, but ICTV and the HPV community revised the protocol for HPV typing, and now whole genome is being used. So we are in the process of launching this server. I have shown here one publication, but we have 3 papers so far from this project. Next. And then coming to immunoinformatics. So these red, so this is, by the way, E6, one of the oncoprotein of HPV virus. The accessible surface area is shown, different epitopes are shown in respective colors, and these red surfaces indicate the mutations that are observed in Indian population. And as you can see, a lot of mutations are happening, are observed in the neutralizing epitopes, and we are then trying to correlate. And next, please. So if the vaccine is developed using the vaccine strain, then and even if the vaccine is administered one of the most recent strains having such mutation, there is always a possibility of escaping the immune response of the neutralizing antibody. So we came up with this informatics-based protocol for development of therapeutic vaccine, wherein we thought that we can look at the allele diversity in the population. So our study population for this particular project is Maharashtra, and we have compiled a set of MHC1 and MHC2 allele diversity. Then we looked at the viral population, and we predicted the epitopes. We identified the variations that are part of these epitopes. Then we carried out molecular docking and simulation studies for those HLA alleles where 3D structures were not available. We predicted them using homology modeling. I'm showing you snapshot of docking of peptide in the MHC peptide binding groove. Beautifully, the peptide docks in an extended conformation, and then we evaluate it. So this is completely done in silico, and then Dr. Madhuri Thaka's group in Nari, we synthesize this peptide, and they are actually carrying out experiment, and we have enrolled a set of patients to see, basically PBMC related analysis to see what kind of response do we get. So we have, let us say, the peptide and the mutant peptides, and we are trying to profile these peptides against the patient population. And we have at the back end these MD simulation and docking simulation studies, and the scores we are trying to correlate with the results that we have obtained in the lab. Of course, this project is not yet published. Next, please. All right. So while we were working on all sorts of interesting projects, it was December of 2019, and on the Twitter I read about this news of SARS-CoV outbreak, and then ever since the first genome sequence was obtained in the first week of January 20, we have been tracking the sequences, and we did this analysis from January to February and April 2020. This paper was published where we tried to look at how SARS-CoV-2 has evolved and learned to jump. So we used the RDRP gene, and we analyzed a set of beta coronaviruses using evolutionary dynamics and population genomics approaches, and came up with this first explanation where we said that the bat, while there are so bats, basically this work showed us that bats are brewing a population of large number of viruses which are likely to jump, and from bat to human transition might have happened through series of other species. So with this we came up with this first explanation pertaining to the spillover of bats, and the purpose for which in 2015 this One Health concept was proposed, and it was exactly said that we have to look at animal-human interface for the spillovers in time to come. Next, please. And then we have been following the literature as part of my role as a editor in infection genetics and evolution journal. We as a group of editors reviewed the progress for SARS-CoV-2 evolution, diversification, and emergence of lineage after an year, and we came up with this particular review. Currently we are working on 2 more projects with respect to SARS-CoV-2. One is sponsored by Serum, it's under non-disclosure, and recently we have carried out analysis of some of the Indian isolates. That paper is under review. Next. All right. Starting with conformational epitope prediction to application of immunoinformatics for various translation projects. Our understanding with respect to antibodies and how antibodies recognize antigenic surfaces evolved, and this very, very beautiful illustration on the left, it's called a broken rainbow. And here you see color pebbles at the bottom of the rainbow. So using this, Dr. Van Regenmortel came up with this concept of antigenic continuum, that we look at epitopes demarked by various antibodies, but the surface as a continuum is available, and then the antibody specificity, polyspecificity, reactivity, cross-reactivity concepts have undergone tremendous review, and we have very, very elegant and fine understanding with respect to antibody specificity. We thought that probably this is a time to look at antibody type specific epitope mapping. So far most of the algorithm, barring the ones which are developed by Raghava, most of the epitope prediction algorithm, they predict the epitope to be antigenic region or not. Okay. But a single epitope can be recognized by different types of antibodies, classes of antibodies, and there was no algorithm available, and we thought probably now is the time to come up with such algorithm where we apply machine learning techniques and come up with prediction of antibody specificity. So we developed very, very recently. This paper was published just last month. Next, please. So AbCPE. So I have given the algorithm here, but looking at the time, I will not get into the details. So you are most welcome to read our paper in front years. I have given the URL there. We tested, what is interesting is that, so here, we predicted the epitopes binding to individual antibody types. There are 5 types of antibodies, as you all know. We curated data from IEDB database for 4 classes. That is the one class for which we did not get enough data is IgD. So IgD is not included in this prediction. And then we looked at combinations of antibody types like IgE and IgA, IgE and M, and so on and so forth. And then I have given the data set that we curated. And in the last column, there is also a data set pertaining to SARS-CoV-2, because already the antibodies have been characterized. Most of them are IgE type, IgM type, but then there are some epitopes which are recognized by more than one antibody. And we thought that beyond benchmarking the performance of this algorithm, we can also test it for the class specificity for SARS-CoV-2 data, and this algorithm works really beautifully. So we got very good results, and I would encourage you to use this algorithm. The input is simple. You need to provide linear B-cell epitopes, either experimentally validated or predicted using any algorithm of your choice as an input to AbCPE server, and AbCPE server would give you potential antibody types that would recognize individual epitope. So that's the algorithm in nutshell. Next, please. So here we are, before, back please. Yeah. So what I shared with you are the development that are taking place, or that have shaped up in my group, and I have had lot of collaborations with my colleagues from the department as well as various other departments. And in general, in the virus bioinformatics space, we are developing databases, algorithm servers. We are collaborating with virologists, and since it's a multidisciplinary area with statisticians, computational biologists within and across. Some of the translational aims that we look at is to development of vaccine or antibody therapeutics. We are recently awarded this center by DBT for virus bioinformatics, and the activities under that particular initiatives are launched very recently. Next. You can go on clicking. So these are some of the databases and the servers. I did not talk about, IRES prediction server, where we predict the internal ribosome entry sites for cellular as well as viral sequences. Next. I think a couple of slides have this animation. Next. So this are just the screenshot where our papers are and where our servers are. Next, please. We have ported these resources on this cloud platform of Pune University. I have given the URL, some hit statistics for some of the databases and servers that I described are also here. So next. So this whole journey was made very interesting because of the people who were part of these projects. So we have over the years, large number of collaborations with the scientists from India and abroad. Virologists I have shown in blue color. Large number of masters and PhD students worked with me in the process of designing, developing, and applying these technologies for the translation purpose. We were generously funded by DBT, Department of Biotechnology, Government of India. We had grants from DST. We had Indo-Norwegian funding for last 12 years for our group. Several JRFs being BET supported and UGC supported. JRFs worked in our lab. I have got a couple of fundings from Serum. So basically, it is credit of these wonderful individuals that I got to present some of the interesting results that we have obtained over the years. And before I really close, I would like to do some crystal gazing and say that what are the opportunities and challenges in this area. The opportunities are enormous. You know, COVID-19 itself pushed our limits and accelerated the pace for data analytics and discovery. So there are ample opportunities. So informatics is easing out the process for discovery at one hand. Second, in slightly different scenario, it is also helping us to use data as a probe to explain how things work and how things don't work. For example, what I have shown in case of MMR vaccine and the Serum case study, or the therapeutic vaccine that we are trying to develop. But one of the challenges in this area is availability of experimentally validated negative data. So this particular field lacks from a very good compilation of experimentally negative data, of course there are some initiatives, and as the data becomes more and more available, the rules for the prediction would be better refined, and with these algorithms would gain higher accuracy. Yet another question that was thrown on face by Raghava in ResearchGate is, we are developing these resources, and where are the applications, who are using? So Raghava, I guess you got a lot of answers, but one of the answers could be, let us put our hands together and say that we try out some of the translation ourselves and show it to the community that the products can be developed out of immunoinformatics, and they do work, and in that direction we are making our own small attempt towards the development of therapeutic vaccine for HPV virus. And this is where I would like to stop. Thank you very much for the opportunity and for being here. Thank you.
Thank you a lot.